Hello everyone, this is your instructor Kwame Shen, and this is part 3 of our lecture on microbial genetics. For this lecture, we'll be looking at regulation of gene expression. So in terms of objectives, we'll look at the constitutive and regulated expression of genes, operons, inducible and repressible operons. And then for the last part of the lecture, we'll look at mutation and genetic transfer. So for gene expression, genes may be expressed constitutively. And what that means is that these are genes that are constantly expressed. So relatively speaking, they are always being transcribed and translated into proteins. So generally we call these genes housekeeping genes, that they help maintain cellular activities and keeps living organisms going generally. So if you look at metabolic process like glycolysis, and for those who are aerobic organisms, you will look at a Krebs cycle and the likes. They will continue to take place, and that they need the proteins in terms of the enzymes to keep it going. So you have constitutive genes that are constantly being expressed and translated into proteins to govern these reactions. And then you have regulated genes. And so these genes are expressed based on the circumstance, based on conditions that are present in the environment, in the cellular environment. They may be induced or they may be expressed based on the needs of the cells. And so the process of gene regulation governs transcription and translation. And so in the end, it controls when proteins should be synthesized and what kind of proteins should be synthesized. For the process of transcription in prokaryotes, you have a double-stranded DNA, you have a promoter region where RNA polymerase is attaching. Remember, you're going to make a messenger RNA in the process of transcription, and so you need RNA polymerase. Now, for a lot of microorganisms, a lot of prokaryotes, there is a factor, there is a protein called sigma factor, which is needed for the process of initiation. So that's the sigma factor that you see, the yellow structure that you see there is a protein which plays a crucial role in the process of initiation. So when the polymerase attaches to the promoter region, without the sigma factor, the process of attachment will not be complete, and initiation of the process of transcription will not proceed. So you have the sigma factor there, and that initiation in terms of RNA synthesis would then begin. But it's only needed for initiation. So after the initiation has been established, it would then leave. Okay, it would then leave, and then the process of elongation will continue until it gets to the point of termination. And after termination, it will then come and bind again to the RNA polymerase. So that's one way of regulating the process of transcription. And so that brings us to operons. Operons are a series of genes that are transcribed together and are controlled by a single promoter. Okay. So a set of genes, as we have here, you have a set of genes, gene 1, 2, and 3. They are transcribed together because they are transcribed into messenger RNA, and they are all controlled by a single promoter. So that's an example of an operon, and you find them in bacteria. They have been studied extensively. So we have two types of operons. You have the repression or repressible operons and the inducible operons. So operons can be repressed or they can be activated. So a repressed operon will have a molecule that will attach to where, next to where the promoter region is, where the polymerase binds, and prevents the process of transcription from taking place. Or you would have a molecule that would attach to the polymerase behind it and activate it, cause it to start the process of transcription. So an operon can be repressed or it can be activated. And so we'll talk about one particular operon in detail. That's the lac operon. Jacob and Monod, these were scientists that did extensive studies on a catabolic process involving lactose in E. coli. Generally, bacteria would go for glucose as a source of their carbon. Most bacteria would do that. But in the absence of glucose, lactose is sometimes used. And so for E. coli, lactose may be metabolized to generate glucose and eventually generate energy for cellular activities. And to do that, you would have an operon that has to be induced, and that's a lac operon. So you have a lactose operon which has to be induced. So the lactose operon is an example of an inducible operon. It only becomes activated in the presence of lactose. So if lactose is not present, lac operon is not turned on. So when lactose is present, lac operon is turned on. The genes are then transcribed and then translated, and then the products that it produces are then used in the process of digestion. So you have these three genes, LAC-Z, LAC-Y, LAC-A. These are structural genes. There is also LAC-I, which is a regulatory gene, but we just concentrate on these three. And you need them to cleave or break up the lactose into glucose and galactose. So what it means is that lactose is a disaccharide. It has to be broken down into these monosaccharides. And then you will need LAC-Y. The genes will be needed to be transcribed and translated into proteins that will be used for the transportation of lactose into the cell. Okay. So these are the conditions that are needed for the lac operon to either work or not work. First of all, you need a catabolic activator protein. So catabolic activator protein. When the catabolic activator protein is attached, so in this instance, this is the catabolic activator protein. It then activates the operon. The operon can be activated if lactose is available. If lactose is not available, the operon cannot be activated. It becomes repressed instead. So let me just walk you guys through this. So in the presence of lactose and absence of glucose, so lactose is present, glucose is absent. Since the organism would need to synthesize glucose and then be able to metabolize it for its cellular activities, you need the enzymes that can actually metabolize the lactose. And so when lactose is available and glucose is not available, the lac genes are expressed because you need the enzymes to actually break down the lactose. However, if you have a lot of glucose available and you don't even have lactose at all, then there's no point in expressing these genes. So it's actually repressed. You have a repressor protein that attaches and prevents the transcription of the genes. However, if you have an instance where there is also no lactose and then glucose is very low as well, it is also repressed. So this is repressed. I would say this is partially available, okay, because it's very low, so it's partially available. And this is high, glucose is high. In the instance where you have lactose available and then you have glucose also available in very high concentrations, then the cell would actually want to depend more on the glucose. But since lactose is available, remember the lac operon is induced or is activated when lactose is available. So if lactose is available and you have high glucose, the lac genes will be expressed, but at very low levels. So it's important that you understand the different conditions and the results, I mean, the effect that it has on the lac operon. We also have the trip operon. The trip operon stands for the tryptophan operon. The tryptophan is a kind of amino acid, the type of amino acid that cells of certain bacteria would need. And this is an example of a repressible operon. It's an example of a repressible operon. So when tryptophan is available, the trip operon is not turned on. Okay, it's very different from the lac operon. For the tryptophan operon in E. coli, when tryptophan is available, the trip operon is not turned on. It consists of 5 genes. So you have 5 genes that are transcribed together, and they are regulated by a single promoter. So when tryptophan is present in the environment, the genes for tryptophan synthesis would not be turned on. Now, after the messenger RNA has been synthesized, because for the trip operon and the lac operon, the goal is to prevent the synthesis of messenger RNA. But if the messenger RNA is already synthesized, there are still modifications that can be made. And so there's post-transcriptional control. So the cell synthesizes the messenger RNA and realizes that it doesn't really need it any longer. What it can do, and this happens mostly in eukaryotes, where you have what we call the microRNA. So the cell will then, the DNA will then be transcribed into a microRNA, and the microRNA will have complementary bases to the messenger RNA that is already present and which the cell wants to regulate. And once it attaches to it, it's able to degrade it. So it degrades the synthesized messenger RNA. Very, very, very fascinating. For bacteria, this also happens, very similar, but instead of microRNA, you have what we call short-RNAs. And the short-RNAs will then help the bacteria to cope with very difficult situations. And so you have a messenger RNA, which the cell doesn't want to translate into proteins. What it would do is that we'll have these short RNAs that will bind with the ribosomes. So it will bind with the ribosomes and prevents the pairing of the ribosomes for the synthesis of protein to take place. So you have an instance where you have these short RNAs that will bind to a particular spot of the messenger RNA and repress the process of translation. There are, however, instances where you have these short RNAs that are attached to a segment of the messenger RNA and actually degrade it. So these controls are all built into different organisms, and it helps in regulating when and how much and where these proteins should be synthesized. So this brings us to the end of our third lecture on microbial genetics.